Let me introduce myself. I am a man really without religious faith. And so why am I here? I was asked by these very nice people, and I said I really should open myself to people who might approach a question differently than I would. I try to approach questions as much as I can from a logical and scientific point of view. And so I'll be interested to see where discussion goes. I, however, feel very strongly that questions with a moral dimension are questions that have to be dealt with on their own terms. And it is one of the most difficult tricks to establish a moral code for oneself without a religious background. Many people feel that it can't be done, that the only source of moral truth is through religion. I don't believe that. And so what I'm going to talk to you about tonight is things that I've done over time to try to satisfy my belief that we do have to take a moral stance. And in my case, through worrying about the moral status of scientific issues. So I began life as a virologist. I was interested in participating in what was then a building revolution in molecular biology, and the simplest organisms to work with were viruses. And so I turned to the simplest organism to begin my career as a graduate student. That was 1960. And many of you will remember that in 1953, not remember from having been there, but remember from having heard about. In 1953, Watson and Crick published a one-page article in which they described the structure of DNA. And that totally revolutionized the science of biology because the implications of it, and the implications were very obvious, were effectively that biology had become an information science, not an observational science, as it had been for so long before that. So I went to work on how viruses duplicate themselves, and in the process discovered that there was a class of viruses that reversed the flow of information that Crick in particular had specified, that is that DNA made RNA made protein. And we found that in the class of RNA viruses, the RNA was copied into DNA. Since that time, that's turned out to be a widespread process in biology. That's a famous picture of Watson and Crick looking at a model, and it was basically the construction of that model that allowed them to determine the structure of DNA. Without the ability to model it, they wouldn't have seen the problems that many other people had run into in trying to understand the structure of DNA. So it's been, for me, 60 years of doing research, of investigating basic questions of life, but fundamentally making biology into chemistry. Some biologists are embarrassed by that. It was just basically doing good science. And those who had a look into the future, who had the sense of where the future might be going, realized that if we could manipulate life in effective manners, that we were going to run into questions of just how far we wanted to go. And that those questions were going to challenge us. And in fact, they have challenged me and all of my colleagues who date back to that time. And there's a whole series of controversies that have occurred. From 1953, when the structure of DNA was first announced, published, for the next roughly 20 years, we had to learn how to cut DNA up, how to paste it back together again, how to isolate it, ultimately how to sequence it. None of that was particularly controversial, and much of it was really very beautiful molecular detail. But when you put it all together, as Conan Boyer did in 1974, they showed that you could make chimeric molecules of DNA, that you could take DNA from a fish and DNA from a bacterium and put them together because it was just DNA. And all DNA is fundamentally the same. And then you could propagate that DNA in bacteria, and that propagation enabled you to make large amounts of individual genes effectively. Something we do now routinely, but was a great accomplishment at the time. And when we looked at where we were, we realized two things. One thing was that the techniques that we now had were going to answer almost any question you could imagine in biology. It was going to take time. It has taken time. But I think we still feel that there's no limit to what we can learn about how living organisms work. And the other side of the chimeric molecules was that they might be dangerous. You might, for instance, carry a cancer gene into a bacterium, and then that bacterium might get out of the laboratory and infect people or animals and transfer its gene and cause cancer. That was a serious concern. And a whole bunch of other worries came up, and I'll talk a little more about that. So that was the first time when controversy hit biology. We had to respond to it. And I'll talk about that response in a minute. And then I've just lined up here on the next slide a whole lot of other things that generated controversy. One of the first was in vitro fertilization, because that meant you could now carry out the process of creating a child in a test tube. And test tube babies were the words that that were used. There were many people who opposed it, many people who were afraid of it. Today, there have been something like 5 million people born through in vitro fertilization, and many, many families, people in this room, I'm sure, owe their existence to in vitro fertilization. But what it did was to open up the process of creation of a human being to manipulation. And I don't think we saw it at the time, but I'll come back to it. And in 1989, the first knockout mouse was made, in which a single gene was altered. People won Nobel Prizes for almost all of the things I'm talking about. That really was gene editing, but it was done in animals. It was not something you would do in humans at the time. And then about 10 years later, Dolly the sheep was born. Dolly emerged from an egg cell that had had an implantation of a nucleus from the mother's, one of the mother's somatic cells. And that was cloning. Now Dolly the sheep had the same genetics as its mother. And there was a furor over cloning. And was human cloning possible? And in fact, human cloning has turned out to be extremely difficult and hasn't been done. But in principle, it could be. And then we learned how to grow human stem cells, cells that each one of which could be made into an organism. And sometime later, Yamanaka and some famous experiments showed that you could induce any cell in the body effectively, almost any cell, to become to grow and have the properties of a stem cell. And so now we had induced pluripotent stem cells, which opened up the possibility of doing human medicine through alteration in the laboratory of cells. But the thing that brought it down to the most effective procedures, and which is in the process of altering everything we think about how to do biology, is CRISPR-Cas9, a method of altering genes. And I'll spend some time on what that means. But we're still creating challenges to the society. For instance, there was a general agreement among scientists and among nations that you could use in vitro fertilization to generate an embryo, but that you could not keep that embryo alive in a laboratory beyond 14 days. Now, 14 days turns out to be almost arbitrary. But it was also, at the time and up until very recently, a barrier that we couldn't even breach, because embryos would die earlier than that. They had to be implanted in a uterus much earlier if they were going to grow into a human being. And so the 14-day rule was convenient. But in the way of scientists, we recently discovered, not we, but people, how to keep embryos for longer than that. And one of the scientists who's sort of at the heart of a lot of the controversy about now how long do you keep an embryo, a human embryo, alive, is joining our faculty in the fall. So we'll be a center of this kind of work. And somewhere around 2000, we began to hear about synthetic biology. Synthetic biology being the use of, the alteration of biological abilities to create new opportunities. And those alterations could be pretty remarkable. We could change the genetic code. We could put new amino acids into proteins. We do a whole lot of things. And I say there, it was started in the 2000s, but it will go on forever. And I can assure you that if you were alive in the year 2100, you would see, and thereafter, you would see remarkable use of biology. And it really is just a matter of how imaginative you are in thinking about what might be possible, because we'll be able to do almost anything. So we're really, even with the things I've talked about here, we're just at the start of a revolution of modifying organisms, and maybe humans, in a sense, to order. So let me go back to what led to a very famous meeting about how to deal with the oncoming revolution. And it's a story about biologists taking responsibility for introducing a disruptive technology into the society in a way that would be acceptable. So in 1974, I said the first chimeric molecules were made. And that work was announced at a summer Gordon conference in New Hampshire. And the first thought that you could propagate what might be dangerous chimeric molecules, plasmids that would grow in bacteria, ones that might have toxin-inducing genes or might carry antibiotic resistance, things that could really interfere with or create medical problems. And the people at that meeting, hearing that this could be done, said, we've got to stop and think about this. What really is the danger? How do we control it? Questions that had never been asked among molecular biologists. And the organizers of the meeting, Maxine Singer and Peter Saul, agreed to send a letter to the National Academy of Sciences. And it was printed in Science magazine. And it warned of the danger that it warned of the concern that they had, that there might be danger. We are writing to you on behalf of a number of scientists to communicate a matter of deep concern. Several reports indicated that we now have the technical ability to join disparate DNA molecules. And we might create molecules that had unpredictable behavior. And they asked, and I've just highlighted some of the sentences there. They asked that the National Academies establish a study committee to consider this problem and recommend specific actions. And so a group of us got together in my conference room at MIT and said, how do we respond to this? And we decided that, first of all, we should ask the community not to do any of those experiments that seemed on the face of them potentially dangerous. And secondly, that we hold an international meeting that would bring together the experts in this area to assess where are we today, where are we going, how do we get there? And so we asked for what has been called a moratorium, although we were careful not to use that word. But the press used it. Continually. And it in fact held. Nobody tried to do the experiments that were outlined, and I'm not going to go over what experiments they were. And we set about to organize a meeting. And that meeting was held at Asilomar overlooking the Pacific, one of the most beautiful places on earth. If you haven't been there, go there. And that now has become called the Asilomar meeting, although there are meetings there every weekend, or every day probably. It was a meeting of scientists, mainly bacteriologists, some of whom had never talked to each other before about where their science was at. And we also had government officials there. We had some lawyers. They scared the hell out of us. We had ethicists, and we had a few professional journalists. And they agreed not to write anything until the meeting was over. It was a 3.5-day meeting, which is a very rare thing, but worked out extremely well, because by the time they wrote their reports, we had come to a conclusion, and they weren't able to influence that. And we consciously limited the kinds of questions that we were thinking about to questions of what were the dangers that might occur and how do we avoid them. And we didn't raise moral and ethical questions. We didn't raise questions about biological warfare, which is in a sense a moral question. Because we were trying to keep it to what scientists could talk to each other about. This was a picture taken of the organizing committee meeting at Asilomar. Sidney Brenner, who's in the middle there, just died the other day. And that's just to prove that I was there. Shows you. Shows you what happens to you over the years. That was my hippie phase. And in the last night, we actually sat up all night trying to balance everything off. We came out with a recommendation the following day, which was adopted at the meeting. And it was effectively that experiments that were very dangerous should not be done. And we keep that in place. But that experiments that on the face of them seem to have no real danger in them should be done. And that we should keep track of whether any problems arose. And in fact, what we laid out there was the basis on which recombinant DNA technology was established in the United States and in the world. Even though there was no international body to insist on any particular rules everywhere in the world except in the Soviet Union, it was honored. The Soviet Union started a biological warfare program which was totally secret and very extensive. Post-Asilomar, and I must say never used either. The guidelines, there was a set of guidelines, and they were, as I said, followed. We had very little experience to go on here. We were inventing it as we went along, but it worked. Now, ignorance in general leads to fantasy. When we see something happening in our society, and we don't know what's causing it, like for instance, when AIDS first was identified, and no one knew where it was coming from or what was going on. The virus hadn't been found. Fantasy reigns. And so, in certain places where there are intellectuals, they can think of really bizarre fantasies. And the major place was in Cambridge, Massachusetts, around MIT and Harvard. And they virtually shut down recombinant DNA research at Harvard and MIT until a group of us managed to bring some sense and order to the situation. And in fact, what we've learned was that the guidelines that we set up were overly strict. A whole lot of that was not necessary. But it was an orderly procedure, it was followed, and it gave the public the belief that nobody was pulling the wool over their eyes. People were being open. The word transparency, used as it is today, wasn't used then. But basically that we were being transparent. And I'm not going to go into the wonderful things that we did, but we revolutionized cancer research. We revolutionized basically everything about biology. And one of the positive things was that we had those reporters there. They understood what was going on. They wrote very intelligent articles. And that began the education of the public about DNA. So before, this was now spring of 1975, before that time, DNA was something that was only known to people working in the field. Soon it became simply the filler in crossword puzzles. And today we debate not only about DNA, but about embryo research and synthetic biology, about gene editing and all of the issues that have come up. I am actually the co-chair of a committee of the National Academy of Sciences called the Committee on Science, Technology, and Law that highlights new developments and brings out the issues as best we can, encouraging debate. We expect the public to care about what we do, and we try to open ourselves to the public. And that was all set in motion at Asilomar. But Asilomar was a mode of expert decision-making. And we're really in a different situation today. Because the questions that we have about embryos and about gene alteration are not questions about the technology. Nobody argues that we shouldn't be able to do the technology because it's dangerous. The question is, should we do the technology because it might not be right? And that's a wholly different question. So the questions that we face today are religious for those who believe, ethical and moral questions. And they're not questions that scientists can answer by themselves. So the process for deciding answers has to be much more public than it was at the time of Asilomar. And we'll now turn to talk about gene editing. We had a meeting about that, and I'll get there in a moment. But now we had not only representation from the scientific community, but from the public, from a number of publics. A start towards a post-Asilomar process. So, in 2013, there were a couple of papers published in Science magazine announcing that a natural constituent of bacteria, which is sort of a bacterial immune system, could be adapted for use in higher cells, in mammalian cells, vertebrate cells. And that it would target changes in the genome to very specific places. And the resolution that you could achieve is the resolution of a single nucleotide. And that is the ultimate quantum of biology, is the single nucleotide. You can't get any smaller than that. I mean, you can get into atoms and things, but fundamentally, in terms of the information content, you can't get any smaller. And so that actually completed the revolution that was started by Watson and Crick when they determined the structure of DNA. It now didn't look at all like Watson and Crick. Two wonderful women, Jennifer Doudna and Emmanuel Charpentier, did the work in the biochemistry of CRISPR-Cas9, and Fang Zhang and George Church adapted it to higher cells. So let me talk a little bit about how you do this, what it enables, what we can expect. So how does it work? The key to altering DNA inside of cells, making a gene alteration, or an alteration in the intact genome, is to make a clean break in the DNA across both strands. And the reason that that is so effective is because nature abhors a cut through the chromosome. And there's a whole army of proteins that are designed to repair a break. And in the process of that repair, you can make alterations. They sort of come along free of charge. So if you can cut DNA in a specific place, then you can establish the conditions for alteration of whatever gene is at that place. So you need three things for this. You need a protein that can cut DNA, a nuclease. You need something that will guide that protein to a specific spot in the genome. And then if you want to heal the break and make an alteration in sequence when you heal it, then you need a guide DNA to specify the altered sequence. It's sort of neat. It's a collaboration of DNA, RNA, and protein. And it's the RNA is CRISPR. I won't tell you what that means because I can never remember. And it bears no relation to what it actually does. The protein is Cas9, and the guide DNA is a little piece of DNA that you put in one of a number of ways. And so this is a chromosome down here. And the sequence in the chromosome is highlighted in blue. And that's the target that we want to find. That's the gene we want to alter. And what finds it is CRISPR RNA. And that CRISPR RNA binds to that site using, and most of you I'm sure will know about this, the standard Watson-Crick base pairing structure. So this is all about finding sequence in DNA. But what's finding it now is an RNA. And that RNA is held in the eminence grise in the background, which is Cas9. It's held onto Cas9. And it finds this sequence through the agency of Cas9, opens up the double helix of DNA, leaving a single strand down below in an RNA-DNA hybrid. And then it cuts right there. And there's specificity to that I won't go into. And this is just another picture that I took from literature, stole from the web, that shows that. And when the cut occurs, you now have a double-strand break in the DNA at that one position. And two things can happen to that. One thing is that it gets healed with random sequence at the junction. And that just inactivates the gene, screws up the information in the biology. That may be what you want to do. And the other thing is to take that donor DNA and implant its sequence right there. Homologous recombination. And that can lead to as precise an alteration as you wish, a single nucleotide. And that's been done, it's being done, I'd say, in half of the biology laboratories at Caltech right And so what can CRISPR-Cas9 do? Well, it can alter any one of the roughly 20, 25,000 genes in our genome, or in the genomes of animals, experimental animals. It can correct a mutation back to what is the standard sequence. It can add a gene, because that donor DNA could actually bring a whole new gene in. It's being done extensively in plants today. We call it gene editing or genome editing, and it can actually do other things. For instance, if you inactivate the nuclease ability to cut, so it doesn't cut, it'll bind to a specific region and sit there. And if you alter Cas9 so that it interacts with other proteins, it can bring other proteins to the DNA. And in that way, turn genes on and turn genes off. Again, with remarkable precision. And this was just found in one bacterium, but many, many bacteria have CRISPR-Cas complexes that are used, as they said, as a sort of bacterial immune system to knock out viruses that may attack the bacteria. And so you've got a huge reservoir of possible proteins that can be used and tricks that are built into them. So it's not just these two. And this is what we really would like to see happen. So, and it takes us back into the history of Caltech. When Linus Pauling was here, he recognized that there was a disease of hemoglobin called sickle cell disease that was being caused by a change in the charge of the DNA. And that because it was a charge issue, you could see that the protein was altered. And he brought it down to a single amino acid change. We now know it's a single nucleotide change, an A to a T. So we could create a way of going from T to A. We could get rid of sickle cell disease. Now we have to do that in every cell in the blood. But we have ways of doing that. And this is sort of the holy grail. There are now many companies trying to do that. And it will be the ultimate triumph of gene editing. And there are lots of genes like this. There are literally thousands of genes caused by small alterations at localized regions of the genome. Call them Mendelian diseases. And so we could, in principle, fix any or all of these and bring them back to normal, to the sequence of the general population. And since we know that that sequence is built in, evolution has built it in to how normal function occurs in living organisms, changing from the mutant to the wild type is not very controversial, except for one reason that I'll come back to. Now, you really can't do this very effectively in an adult organism or a child that's been born, because we have trillions of cells in our body of any particular kind, and changing a large enough number of cells to reverse diseases is difficult. However, we all came about from the union of an egg and a sperm. And at the time of that union, the whole organism is held in a single cell. And so we can inject into that single cell CRISPR-Cas9 and modify its genome. And that's in fact what we're talking about. That's the most exciting and difficult and scary scenario. Now, we may be able to actually, and I'm not going to talk about how, get access to the precursors of eggs and sperm and make the modifications in those precursors so that the eggs and sperm are corrected from mutant to wild type. And that may in fact be the only way we can use this technology for reasons that I'll come to in a moment. So this is germline therapy, and it would be passed from the child that was born to its children and down through the rest of evolution. And the important thing is, first of all, that many people who carry mutations want to have children that don't carry those mutations. And secondly, the only way to use this technology, at least at the moment, requires in vitro fertilization. So in vitro fertilization is pretty straightforward. We take eggs from a woman and sperm from a man and put them together in a petri dish. And you can see there the sperm attaching to the egg, or you can inject the sperm into the egg if you wish. And that gives rise to a zygote. A zygote has the nucleus from the sperm, the nucleus from the egg. So it actually has all of the genes of the organism in a single copy. And that divides and becomes a 2-cell embryo and a 4-cell, an 8-cell, and goes on to develop to a blastocyst, at which point it is, in vitro fertilization, put back into the mother's uterus. But you can superovulate a woman and get a lot of eggs, and you can obviously get a lot of sperm. And so you can make a large number of potential organisms from a single mother and father. And if the genetics works out right, and I'm not going to teach a course in genetics, some of those embryos are wild type, and others have mutant genes in them. And for a very simple reason, you can find out which has the wild type genes. And that is that these are 8-cell stage embryos. And you can take a single cell out of that, or a couple of cells, and the rest of the cells in there will go on to form a normal animal, person. So if we could find out which embryo has only good genes, that would solve the problem. We don't need to modify genes. And so let's say we did that, and we found 3 good embryos, and they would then be implanted. This is called Pre-Implantation Genetic Diagnosis, PGD. And PGD will find good embryos. There are many children being born today after PGD. It works for dominant genes, it works for recessive genes. It won't work if both mother and father have mutant genes, two mutant genes. But that's very rare. Now, if this worked 100% efficiently, the need for gene editing would be pretty small. But it doesn't. And in many cases, there are very few eggs available for modification that they don't take very well. They estimate in one round of in vitro fertilization, there's about a 25% success rate, which is not what you hope. And so CRISPR-Cas9 could change all that. And that's probably the biggest use of CRISPR-Cas9, is to be put into the zygote to modify a gene so that there are now well-defined embryos that have only wild-type genes. And the problems of that are, first of all, that it all depends on forming a double helix between RNA and DNA. And you can easily imagine that there could be other places in the genome that have a sequence so similar to the sequence at the site you wish to modify that you get modification at the wrong site. And you get cutting at the wrong site. And so off-target is a big issue. How good are these CRISPRs? The second thing is that you can get mosaics, because as those cells divide, if CRISPR gets to work in one of the daughter cells, one of the later stages, and modifies one cell, then no longer are all the cells identical. No longer can you assay the genetics by taking one cell out, because you don't know that the other 7 cells are identical. And that, to my mind, is the biggest single problem with the application of this technology, and why I have alluded to the fact that I think you have to go back and modify at the egg or sperm stage. Now, PGD is being done today, and the gene editing would be sort of added on top of that. Now, this is for rare Mendelian diseases: cystic fibrosis, Huntington's disease, sickle cell disease, lots of them. But there are a lot of gene diseases that we have, like heart disease or Alzheimer's or schizophrenia, which are caused by all sorts of combinations of gene mutations, and a lot that isn't genetic. There are traits that we have, like height, which are caused by 100 different genes, or affected by 100 different. So those would be very hard to deal with. And so we're really talking about the single cell. But people have this idea that we may be able to modify offspring in directions that people want to enhance their offspring, so everybody can go to Caltech. And that's not as true as it might seem in science fiction, because it's so polygenic. But what's really worrying everybody is the slippery slope. That if we start modifying the genes for single diseases, and we get good at it, then there will be a tremendous pressure to make enhancements of the genome of offspring. And that could go on to sort of wholesale modification of the genes of children. So if you're thinking about what the problem is, I think the slippery slope is the problem. So anyway, this led us to have another meeting. It was held in December 2015. It was sponsored by the National Academies of the United States, the Royal Society of England, and the Chinese Academy. I chaired this organizing committee, and the meeting itself took place in Washington, D.C. And it came out saying that we are not ready to begin any modification of the genome of human beings, largely because we don't have the skills to do it yet. We said in particular it would be irresponsible to proceed. And then a more scholarly report was written, and that again said that maybe one day it would be permissible, but right now we didn't know enough. And I'm not going to go through all of that. In 2018, we held a second summit in Hong Kong to see what had changed in those 3 years. And there were many things that had changed, but we still agreed that the off-target effects and the possibility of mosaicism were things that were not under complete control, although particularly the off-target effects are now generally thought to be minimal. And we heard about great progress, mainly from David Liu, who showed how you can alter specific bases in DNA without making a double-strand cut. However, at the summit, one of the people invited to give a talk about his ongoing work announced that he had, in fact, already modified the genomes of 2 children, 2 embryos, and that they had been implanted and the children had been born and were thriving. He's J.K. And at the summit, when he announced this, it was a zoo. First of all, it had been signaled that he was going to do this. The press knew it. Press came from all over Asia. And what he had done was counter to Chinese law. Not only did he countervene what our committee had asked for, he countervened the law, the Chinese law. He's in some kind of a house arrest at the moment. And people are now talking about a more formal moratorium, thinking that they can stop this kind of behavior. I don't think you can. As somebody pointed out to us, we have laws against murder. It doesn't stop people from being murdered. What's more important is that we have to establish a moral position here and hold to it. So my own opinion, and this is the end, is about 2 questions. What will happen and what should happen? What should happen is we need to perfect these techniques. That's ongoing. We should have a global understanding among countries about what's the appropriate way to move ahead. To my mind, that would be to to focus on the single gene defects or single gene mutations. And I think we can do good for families that want to have children that pass down the genetics of their parents. But what will happen, I suspect, is that we will get to the point of being pretty comfortable with the technology. And a physician will be faced with a patient. And I've talked to physicians, and they say this is when they have to make the most difficult decisions. And that patient will say, I want to use gene editing to have a child. And it will be the physician's judgment that he can do that safely. And he will want to do that. And it will produce headlines if he does, and hand-wringing. But if there's agreement at that point that we can do it safely, I think the pressure on physicians will be enormous. So let me wrap this up. Let me point out that the way we've determined the genetics of our children has been a lottery. A few genes from dad, a few genes from mom. Put them together and you get a baby. What we're seeing now is the ability to control this process. It's still rudimentary, it's going to get better and better. And the question that faces us is, do we want to leave our destiny to chance? And many people do. Or do we want to take control? And are we smart enough to do it well? And whatever decisions we make, we have to remember that we're talking about lineages that will then go on forever, or at least as long as there are humans on Earth. Thank you.
We'll have time for questions and answers, but because this is such a large room, we want to make sure that the person who wants to ask a question has access to a microphone. And so if I could kindly ask, if you're interested in asking a question, can I actually get you to go outside and form a line outside the door, and what we'll do is we'll have you come in so that I can hand you the mic. In the meanwhile, can we just give one more round of applause for Dr. So while we're waiting for people to line up to ask some questions, and I highly encourage you all to do so, I'll start us off with the first question. And it relates to why we're meeting here and why SAFE decided to put on this event. And we put us on this event in large part because of that one line you said in your talk, which is that we're at a point where we're faced with a question where science alone may be insufficient to address it. And we need to draw on spiritual frameworks, more philosophical, ethical frameworks. And so along those lines, I wanted to ask, how do you think that scientific bodies can engage in productive discussion with other bodies, including religious bodies, in helping to move this conversation forward in a healthy way?
Well, this is a good example of how that can be done. It's got to be done in relatively small groups. And it's got to be done with a background of understanding of the science. And I think that's how our society makes such decisions, we often leave such decisions to our representative, our political representatives. I don't think we can do that with an issue like this. And furthermore, if you remember back to the decision that we made at Asilomar, that decision was to start with barriers high and lower them over time. Politicians are very bad about allowing flexibility and of changing their mind. So we worked very hard to keep politicians out of the discussion, and we were successful. So no laws were passed, and it was all done by NIH-based guidelines that were had the cloud of NIH had the cloud of being able to cut off funding. Today, actually, we have so many private sources of funding. Even that is not a good answer. So I think the answer is we need to convene more than just summit meetings to discuss these issues.
That's why we're here today.
And that's why I'm here today.
So we'll have our first question come up. Sorry for the awkward walking across.
This is an awkward way to read things. Yes.
Hi, thank you so much for your time. So you addressed a lot of the ethical issues with the technology in the talk, but I was wondering what you think about the consequences of specific mutations. For example, Lou and Anna, it's been speculated that they might have an increased risk for West Nile virus. Do you think that there's a way that we can conduct clinical trials for these sort of modifications without already committing to a particular ethical position?
Yeah, you can do things in animals. In particular, in non-human primates, with the idea that non-human primates are close enough physiologically to humans that we could assess what a given gene does. But we actually know that that's not a good enough answer, because primates, we can't tell what they're thinking. The whole issue of genes having effects in the nervous system are very important and very hard to assess outside of human beings. So, yeah, we need to focus on what a given gene can do. The nice thing about starting with Mendelian traits is that we know that the wild type configuration has been tested in human, it's almost all humans have the wild type. And so I don't worry so much about it. Now, sickle cell is an interesting example because sickle cell is valuable to individuals living in Africa in sort of primitive situations. And so the people who are suffering from sickle cell in the United States are not at risk of malaria. And that's the advantage that you get from the sickle gene, from one copy of it. So you might say, you know, you're taking away an advantage from them. But I dare say that the risk is much higher than the benefit's much higher than the risk in that situation.
Hi, so I wanted to ask, I think there's news recently that the CCR5 gene that JK had deleted, that change could have also improved symmetric neurological function and like stroke recovery. So in general, how would you say we deal with like genetic modifications that both you know, remove, let's say, a disease, but also might confer other enhancements?
Again, it's a sort of risk-benefit analysis, but I certainly think that what he did, what J.K. did, went way ahead of where I would be comfortable. For the fact that there are positives and there are negatives. Last gentleman was talking about the negatives, you're talking about the positives. I don't know how you make that balance, and I think you stay away from it. It's a useful thing to be able to knock out the gene for CCR5 in people who are going to be at high risk of getting AIDS, of getting HIV infection. We can identify some of those people. There we could make a risk-benefit calculation. I think that might make sense. But I think really you just want to make that modification in a somatic cell or in somatic cells and not in germline cells.
Hi. So I have two questions. The first one is also in the case of JK. So in that case, the babies have already been born. We can't really stuff the babies back. So what shall we do with the babies, and what shall we do with the aftermath of that big surprise? That's my first question.
All I can say is I'm glad that I don't have to make that decision. It's a very difficult decision. I think fundamentally, we don't know where those children are, at least I don't know where those children are. And maybe they could be raised without a lot of notoriety. And we'll treat them as just children. They are basically not at serious risk of anything that's life-threatening. So the best thing they could have is anonymity.
So my second question is kind of longer. There is an ongoing effort of sequencing the Neanderthal genome that's called the Neanderthal Genome Project. And so once it's the Neanderthal.
Neanderthal, yes.
Yes. So once that's done, there is a possibility of reviving the Neanderthals after they've died out for like some 40,000 years. And there are already people volunteering to be the surrogate mothers for them. And their arguments are that we are responsible for the extinction of the Neanderthals, so it's our responsibility to bring them back. And the second one is absurd, is that they're saying that we want to learn more about the Neanderthals, and it's good to bring them back so we can do more experiments on them. So I want to know your thought on this. As humankind, what shall we deal with this, or what shall we think of this? Because of course we can't know what the Neanderthals would think of this, but I want to know what we should think about this.
Well, there's one thing you said that transgresses a very important principle, and that is that you don't have a child for the sole reason of using that child as a subject for investigation. That's just simply, I mean, in my moral compass, that's wrong. And I think most people would agree with that. I don't think we can bring back the Neanderthals because it's a huge engineering project. But one day, maybe people will want to undertake it. We have to treat them with enormous respect, as we would any homo that we brought back. And there are lots of, and more being discovered every year, of species of hominids that raise the same issue. Most of them are hard to sequence. Neanderthal is just barely able to be done. You know, they want to bring back the woolly mammoth too. Let them do that.
We'll take our last question before we break into our discussion groups for the day.
Thank you very much for a fascinating talk. A lot of the model that you've proposed for resolving these questions within the scientific community puts a substantial impetus on the scientists themselves to have the skills necessary to be able to frame and think about and come to decisions about these questions. And at the same time, it does seem to me that within many areas of science, scientists do not spend a lot of time thinking about the moral component of their work, and they're not well equipped to do so. Indeed, many people are kind of actively disdainful of a lot of philosophical and ethical inquiry. I was wondering if you, looking at generations of scientists to come, are you optimistic that the scientific community has the tools necessary to confront the profound challenges that we will face over the next century? Thank you.
I don't expect that scientists alone or even working together will provide them the moral guidance for the society. They can be the advisors. They know, sort of right from wrong, or at least true from not true. But they have to do it in collaboration with others. Who are the wisest people in our society? Not necessarily in the United States, but in other countries, judges are often the wisest people. There are, in particular, judges have made a huge difference in South Africa. And it's a place that people of very broad concern make their livelihood. And we need to find people like that. But I agree with you, the scientific community is not the best place. On the other hand, there are scientists of every generation who are willing to open themselves to these questions, and providing forums for them to think about this. And one scientist, for instance, who made a huge difference in the 2 meetings we've had about genome editing is Robert Lovell Badge from England. And he has educated himself well enough to be able to handle some of these questions.
Let's thank Dr. Baltimore one more time.